principal research scientist from the Animal Genetics and Breeding Unit at UNE in Armidale, and really are the brains behind the RBVs and our genetic evaluation. So it's only fitting to have them here today to introduce these new research breeding values for reproduction in Merinos, and talk about the work that's gone behind it and some of the things that will be of interest to you as breeders. We also have a few of the other AGBU staff that have been involved on the call today, as well as we'll hear about Emma later on interpreting the results. So as we go through today's agenda, we'll be pausing at sections to go through questions. So you have the opportunity to either unmute yourself and have those questions answered, or if you type it in the chat box, we'll make sure it gets answered at the end of each section. So that's it from me, and without any further ado, I'll get Em to start sharing his screen and hand over to Kim to start us off.
Can I get rid of this one? Okay, I think we might have aged a little bit between those pictures that Peter just put up and getting these results out. But we're pretty excited too. I think we were asked a year ago how long it would take to get this going for Merinos. And I said probably about a year. It took a little bit more than that. There was a few more hurdles to overcome, but we finally got there, and we're pretty excited to see the results from this analysis. So I'll just break it down a little bit. I'm going to do the overall introduction, and Andrew will talk a little bit more on the genomic evaluation part of it, and then Emma and I will return back to things like effective recording, and I will try and remember to stop, but I've no longer got any video sort of coverage of anyone, so Peter will just have to interrupt me, and I'll wait and see if there's any questions. So these are basically the new research breeding values for Merinos that we're going to talk about today. And I thought it might be worthwhile talking about the motivation for changing these in the first place, because I know that if you're interested in reproduction, you will have been looking at and getting breeding values for NLB and NLW from sheep genetics. And really, when you think about it, those traits have been motivated by production metrics that get used in the industry, to, most typically lambing and weaning percentages, and they're expressed relative to joined ewes. So we know that those metrics are in fact are affected by conception rates, and then we only have a proportion of those ewes which have records for litter size or lambs reared. So we have three things happening there. We've got conception, we've got litter size, and we've got the rearing of the lambs that are born. And the reality is that those percentages, so the lambing and weaning percentages, diverge with increasing litter size. And that's because the proportion of ewes having multiple litters changes with the increasing mean litter size, and also because the lamb survival differs with litter size. So we're trying to capture all of these things with one metric reflected by one trait. I thought it would be useful to kind of demonstrate this. So here we've got a 95% conception rate, and for each mean litter size, I've calculated the proportion of ewes having different size litters and used the survival rates for those litters to show the divergence in lambs weaned as litter size increases. And the size of this arrow basically reflects lamb losses, which is not really evident necessarily when people look at their percentages. If we change conception, then all we do is shift that lambs weaned up and down. But you can see that if we change lamb survival rate, so here I'm making the survival rate for twins and triplets worse. You can see that we can get quite similar weaning percentages, if I can find the arrow, but for two very different reasons. So we can have a yellow line which reflects a lot of lamb losses, and that's this big arrow up here, or we can have a grey line which reflects normal lamb losses but poor conception. So it's pretty important to understand that that metric may not capture everything that's important, and lamb losses are definitely important because they have a very different cost structure to say dry ewes instead of lamb losses, and also they're typically considered to be a welfare issue as well. So knowing what's happening with lamb losses is pretty important. And the way we value selection will also change. So this is an example of litter size increasing again. Can't see my arrow. This is the line of economic value of further increase in litter size. You can see that as flock litter size goes up, it becomes less and less important to actually increase litter size genetically. But up here, it becomes more and more important to keep the lambs alive that were born as the flock litter size increases. So we basically need to have a capacity to select for each of those components independently, and we can't have that if we only have a trait like NLB or NLW. There are some extra problems with having just a single trait reflecting reproduction. And the first is that you can have a ewe who, if we look at these as these little bubbles as representing conception or not, litter size, and then the survival of the lambs that were born, you can see that a ewe can have an NLW equal to 0 for two different scenarios. The first scenario is that she never got pregnant in the first place, and the second scenario is that she lost all of her lambs. So again, different cost structures and different problems with the relative importance of the phenotype. In addition, people apply management at different times to try and optimise their outcomes. So we don't necessarily want to apply the same contemporary group to the outcomes that result from management before joining to both lambing and litter size outcomes. So for example, lamb survival is going to be more affected by how you manage the ewe before lambing, whereas conception and litter size are obviously only influenced by how you manage the ewe before joining. So I hope that's illustrated that actually we really should be looking at several traits, because there's more than one way to get the same NLW and the same percentage lambs weaned. And each of these component traits can have a breeding value, and that way you know what you have actually selected for. You can value the change properly for the different traits, and we can actually use better models and better correlations with other traits to do a better job of actually predicting the reproductive performance for each of those components. In addition to that, we have worked with sheep genetics to get more data from breeders. So this new data actually offers opportunities to expand the reproductive analyses. We've also done a lot of work in development of analytical software that actually produces the breeding values and gives us a bit more flexibility in how we can do some things. And in the industry, we've also got a lot more genomic data than we've had previously. And in the current sheep genetics analysis, we don't use that genomic information for breeding values for reproduction, but in this one we do. So I'll just work through each of the traits that will get reportable breeding values from this analysis. The first one is, did the ewe conceive? So for every joined ewe, did she conceive or not? And we get that information from the lamb tags, including the dummy dry tags that people enter. And we can also get that from pregnancy scanning data if the value is 0 for no conception. But we've actually modified that. We actually put a time limit on how long a ewe is allowed to take to conceive based on use of lambing dates, etc. If a ewe only conceives after a very long joining period, in this case, there's evidence that she's taken more than 90 days to conceive, she can have conception assigned to be 0 because it's not a rapid conception to the introduction of rams. So that kind of change to the phenotype is actually important if you want to put pressure on timely lambing and not having to extend your joining periods to have more and more ewes getting pregnant. And it also means that we don't get a contemporary group where, because the joining was so long, nearly every ewe is in fact pregnant, and so there's no information on what the conception would have been had we have had a shorter joining period. The second trait is litter size. So that's all about how many lambs were born. Now, because we developed this for maternal breeds first, we've taken the stance of having mothering up as the preferred way of getting this data, and that's really because more information can be recorded if it's necessary. That can be things like lambing ease and birth weights, and also because hopefully you're capturing the lamb with its mother very close to birth. But we will use scan data if there is no lambs recorded. There is some debate about which is actually more accurate, and I think for different situations maybe that answer is not exactly the same. But what we're really after with this trait is a trait which represents the ovulation rate of those ewes and the embryo survival. And probably I've underlined ovulation rate because it's really the big driver of litter size. Embryo survival tends to be a lesser contributor. So that becomes a count, and we'll only accept up to 4 lambs per ewe, because typically if there's more than that, it's because there's been some problem with the data. And it's such a low percentage that we really can't use that data anyway. So this is just to highlight that there can be errors. There can be errors in scan data, but there can also be errors in the assignment of lambs to dams at mothering up. The third trait is about lamb survival, and I'll just distinguish a little bit here about why we're not doing lamb survival itself. I think pretty much everybody knows that management and environment have quite a lot to do with lamb survival, whether that's weather events or how the ewes were managed pre-lambing, etc. With respect to genetics, we have both the genes of the lamb and we have the genes of the ewe, which contribute to lamb survival. And typically, if we were going to run a model where the lamb was the animal of recording, we would have a lamb survival analysis. And if we're going to do it at the level of the ewe, we call that ewe rearing ability. In addition to those genetic effects, every litter has a common non-genetic effect. So this is another thing that can influence the observation of lamb survival for different litters. The problem with a lamb survival model, so a model based on the individual lambs, is that often dead lambs are unreported, they're often ungenotyped, and they're often unpedigreed in the data that we receive. So that makes it very, very difficult to actually run an analysis at the lamb level. In addition, lamb survival in pretty much every species is lowly heritable. And in most species where there's a maternal component, a rearing ability of the dam is often about twice as heritable as the lamb survival. So we can get better heritabilities on ewe rearing ability than we can on lamb survival. And we can also record ewes repeatedly for their ability to rear their lambs. And in fact, we don't even need to know the pedigree of the lambs to get records on ewes for their rearing ability of the lambs. So it's a much more robust way to at least start working on the lamb survival side of things. And in fact, if the ewe walks away when the lamb's born, no matter how good the genes are for the lamb's own survival, it's going to struggle to survive. So working on the maternal side first makes sense. So the trait that we will actually do is ewe rearing ability, and that's all about how successfully the ewe reared her lambs. In order to do that, she must have lambs recorded, including the dead at birth lambs, and ideally the rear type is correctly updated on records for every single lamb. We've discovered that that's not occurring as a general rule. So we have another filter in there that looks for evidence of post-weaning survival. So if lambs have any evidence of being recorded later for wool or weight, we assume that they were definitely alive at weaning, but if we can find nothing about those lambs, we assume that they're not. So the rearing ability trait is actually the number weaned divided by the number born. And so therefore it's always in context with litter size. It's just a reminder to say that. So just an example for the rearing ability trait. If we had a lamb survival model, find the arrow, we would have records for individual lambs. This would say that each lamb survived. But if we have a rearing ability model, we have a single record for the ewe, which reflects this, the ratio of lambs surviving to lambs born. So if we have one lamb that dies and one lamb that lives, the rearing ability is half. And if both lambs die, the rearing ability is 0. And we actually accommodate the litter size challenge in the model for rearing ability, because we all know that it's not as easy to rear twins or triplets or higher order multiples than it is to rear singles as a general rule. The other maternal traits we'll include in this analysis are condition score. There's been a lot of talk about condition score in the industry, and also maternal behaviour score, which is basically related to the behaviour of the ewe in response to a human turning up to actually process the lamb. So that's one of the other traits that obviously can be recorded if people are mothering up at lambing. It's not possible to record that if you're not there pretty close to the lambing event. So just a reminder here, a better maternal behaviour score is actually a lower one, not a higher one, because a higher one means that the ewe went further away from the person processing the lamb. So I'll just stop there in case there's any questions.
There's been a lot come through, Kim. I'll read it out for you, and if you want to have a go at answering them.
Yeah.
We've had one from Anthony around, is there any work being done on gestation length ASBVs? There would be a lot of data from AI programs.
So we have done gestation length. We have a lot of information on gestation length in other species. I think as a proportion of all data, the amount of information we have on gestation length in sheep is actually very low, because nobody tends to operate huge AI programs. There might be the isolated place where there's a lot of AI. So we do know that it is related to survival, but as for applicability across a wider industry, we either need to have more AI or we need to have another way of inferring gestation length.
And I think, Daniel, you've done a little bit of work in this space. Did you want to comment on anything there?
Yeah, I think like Kim said, there's been lots of work done on other species, and we've done some work in sheep. It's, we do run lambing ease analyses that include birth weight and gestation length in maternals and terminals. We also do run it, the analysis in Merinos. We haven't reported it to date. We need to do more work in Merinos. I think it's a trait that we should devote more effort to. Our work in research data suggests that lambing ease and birth weight are obviously strongly related to lamb survival, and it's an easy thing we can target to help us. So I think we'd like to do more work, but we do need to get good data, which also needs more work to get it through the system.
Yeah. And it's one of those traits too where you have trouble in both directions. So too short a gestation is problematical and too long a gestation is problematical.
Yeah, good points. Question from Sally, for conception, how do we handle double joinings? For example, being dry for the first joining and then conceiving to say a backup, or autumn versus spring lambing?
So the issue of a backup, if a backup is within an AI program, for example, it is a ewe that gets pregnant within an AI program with a backup shortly after, it would still conceive providing she was in the 90-day limit. The difficulty with repeatedly joining ewes is that you're not comparing animals the same way each time you do it. Ideally, we want to know what is the outcome from the first event or the first opportunity to become pregnant for that ewe, and not for repeated attempts to get that ewe pregnant within any particular year.
Good one. Another one here around if you enter scan data, how can you enter the scan data and then back up or provide actual birth information, and how is that used in the evaluation, Kim?
So you're now talking about performance recording software, and each of those probably have its own foibles. We use the data that's delivered to us. So we have created a situation where you can provide scan data through the mating module, and that data should be unadulterated. It should be exactly as your scanner tells you what the result is. And then different performance recording software can actually be, you can, some you can upload that as birth type and then adjust it later once you've actually mothered up lambs. So the birth type data is adjusted, not the scan data. And other software might behave differently. So I probably should leave it to other people to discuss software limitations regarding performance recording. But I would like to use pregnancy scan data exactly the way your scanner has told you about it, not adulterated or changed in any way once you've actually seen whether they're right or wrong. And then that way we can also provide some feedback on the accuracy of the scanning that you received.
Yeah, that's a good one, Kim. So if you're doing both, where you are preg scanning and say mothering up, if within the limits of your software, submit both, so we can look at that down the track as well for future research.
Yes, yes.
But from the analysis point of view, we'll use the true birth type.
Well, I'll get into that a little bit.
This is a good one around. Oh, sorry, Kim.
I'll get onto that a little bit more shortly, because we do actually check for consistency between the lambs reported and birth type, and some of that will relate to whether people have used scan data or not. Yep.
We've got a lot of questions coming in at the moment. I'll just go through the next two. If we don't get to your question, we'll make sure we write it down and answer it in when we send around the slides as well. So final two, just for this section, how much increase in accuracy do you get if you have a ewe that's recorded more than once for a lambing event? So having multiple.
I think it would be better if we provided that information separately, because that's to do with the genetic parameters for the trait, and we can provide examples of how many repeated records would add accuracy to that.
Yeah.
But it's a low heritable trait, so measuring more is better. I can easily say that.
Yeah. Very good. And final one, just for this session, is how important is maternal behaviour score in calculating ewe rearing ability, which I think we might come to a little bit later too, Kim.
Yeah, we'll come to that later as well. It's used as a correlated trait. So it's not used as a correction, it's used as a correlated piece of information about rearing ability. Yeah, and it's favourable.
Thanks. So those 3 other questions, I'll write them down, guys, and we'll make sure we get an answer to you at the end of this session. And Kim, I'll hand back to you.
Okay. So I'll go through what data we're actually using for the analysis now and how we're using it. And I'll talk a little bit about models and also some other results from the analyses. So the most obvious thing is that we're actually using the new data that we targeted from breeders in the first place, and that includes joining details for all ewes, and that comes with joining dates, information on service size, and information on management groups for joining within that flock. So we haven't been able to have actual joining information before contemporary groups, so this is a really good initiative that breeders have taken up on and are now telling us about their joining groups directly. We also obtain our pregnancy scan data through this mating module that we've worked on, and that's good because now it means that not only do we have a joining group, but also the scan result is tied to the actual joining group, so that if people have gone and joined more than once, we have a better chance of getting the scan result that actually reflects the outcome from that particular joining contemporary group. But having said that, we're still using lamb records to take precedent over scans. So if the breeder reports that 1 lamb was born, but 2 lambs were scanned, we're using the 1 lamb born. And we're obtaining the new traits which breeders are providing this way, which is maternal behaviour score and also pre-joining weight and condition score. And it's kind of useful to have these pre-joining weight and condition scores because we know for sure that weight has been taken when the animal is not pregnant. We're also obtaining a lot of data from correlated traits, and these are the weights, scan fat depths and eye muscle depths. We're getting those from 3 time periods, the post-weaning, yearling and a hogget, and we have to be fairly careful to avoid duplication and also weights that are already on ewes that are already pregnant. We're not recommending that you increase your recording of weights and fats and eye muscle depths over multiple time periods. The only reason we're including these 3 time periods is because breeders tend to use one or another. So breeders tend to have either the post-weaning period recorded well or the yearling or the hogget. They don't tend to have all 3 recorded. So the only reason we've got those 3 groups in there is to allow people who've not recorded in one period to have data in another period. And we only take that data for flocks that have already provided some ewe reproduction data. So we certainly don't take all the data from all of the flocks. We also use the scrotal measures for post-weaning and yearling males. We've done a really massive rewrite of data preparation software to try and detect and filter all the types of errors that can occur. And we actually have built all the filters on a trait-by-trait basis. So instead of having something that only does a fairly general check within flock year, we actually have trait-by-trait checks for contemporary groups within flock years. And we also have tried to ensure that we have the consistent contemporary group across both data sources. So sometimes we have lambs and no scans, sometimes we have scans and no lambs, and sometimes we have both. And that'll work providing people or breeders actually provide the joining details for all of the ewes that are joined. That's the way it works best. And because we can do the filters by component trait instead of by the conglomerate trait, which is NLW or something like that, we have currently about 250,000 NLW records being used in Merino Select. But in the RBVs analysis, this is actually increased to 540,000 litter size records and around the 240, 250 mark for conception and lamb survival. So it enables us to improve the use of the data that's in the system on a trait by trait basis and to make sure that each of the components are more accurate. Some important filters that affect Merinos, they don't affect maternal breeds quite so much, is that we've had to work out a way of actually making the data that we use effective. And the way we do this is every flock year we check to see how many dams recorded on lambs, and that has to be better than 70%. And there's a couple of reasons for that. The first is that the less dams are reported, the less confident we can be in the errors regarding the lambs that are assigned to dams. And in addition, if very few dams are reported, then very few young selection candidates will actually have reportable breeding values that are accurate enough to use. So this is sort of a global filter that just cuts the data back immediately to data that's actually going to be effective for reportable breeding values. The second filter we have is to make sure that the lambs recorded is actually consistent with the birth type reported on those lambs. So this is intended to avoid the situation where the only lambs we know about are the lambs that are reared, and that's a trait that's less heritable compared to the trait we're actually after, which is how many lambs were born, or the litter size, which is a more highly heritable trait. So we have to put that filter in place because in particular Merino breeders tend to err towards reporting surviving lambs, and we can't really run an analysis on litter size or lamb survival if the only lambs that are reported are surviving lambs. And the analysis doesn't include, any outcrossing. So it's basically Australian and New Zealand, Merino and Polwith. The filters for the mating information we've been provided, well, the RAM in date must be present, and we're actually using dates for a lot of cross-checking and validation in the data preparation. The pregnancy scan data, if the data is to be used for litter size or birth type, for example, we're actually only allowing it within a certain date range. So all pregnancy scanners will tell you that if you don't scan ewes in the right age range for fetal age, they can't guarantee the accuracy at the extremes. So we actually we use it to assign conception if it's outside the limits shown on the slide there, but we don't use it for the litter size count. And again, it's secondary to the lambing data. So if we already have lambs recorded properly, we will use that instead. The pre-joining weight and condition score is just limited to 30 days prior to the start of joining. So it's got to be, within that 30-day window prior to joining. We actually separate the reproductive performance into yearling and adult traits. We didn't actually have enough data in the yearling class for rearing ability, and the heritability was essentially 0 in the current data that we have. And that's not to say that it's going to be that way if we obtain more data on that trait. So at the moment, Merinos don't have a lot of yearling data, so we're only providing yearling, fertility and conception, or conception and litter size RBVs, but the adult ewes are getting the full suite. We have weight, but we're not providing a breeding value on weight, we're providing a breeding value on condition score. You have plenty of breeding values for weight from the main analyses. So I'll stop there again.
Excellent. I think Andrew's actually just answered the one question that came through in that session, Kim, but there is probably one or two that were asked previously that was relevant to that. One was from Anne, do you account for the ewe's previous pregnancy/lactation status?
Yeah, can I get to that in a minute, because that's in models.
Of
So
course,
yes,
yeah.
we go. I'll cover that a little bit more in a minute.
Yeah. And one more from Sally around, should we be submitting who the preg scanner name is, similar to what we do for carcass scanners? And I think that's a yes, if we'd like to look at that in the future.
Yes, and I believe there's that capability is available in the software, performance recording software.
Yeah. Very good. There's been nothing else come through, Kim, so I'm happy to keep letting you go unless anyone would like to take themselves off mute and ask a question. Alrighty, keep them coming through in the next section. Sorry.
Okay, so hopefully I'll cover a little bit more on the models in a minute. Firstly, I thought this would be useful to show people how much data we're bringing into the analysis. And I mean, obviously, there's pretty widespread recording of, you know, the early, I'll call them early development traits from here on, because this is how animals basically grow. Here's the pre-joining weight up here for ewes, and you can see we've got about 42,000 records submitted there. But we've got many more submitted for all of the earlier development traits. We've got about, you know, 35 to 52,000 scrotal measurements. And then in the area of the actual ewe reproductive traits, we've got the 220, lost my arrow again, 220,000 or so conception records, 285,000 rearing ability records, and the 542,000 litter size records, and relatively few records so far on yearling information. Now, the data that we're using enables us to have better models on a trait-by-trait basis. So we do actually fit separate contemporary groups for the joining versus the lambing outcomes. So joining affected traits are obviously conception and litter size, and the lambing affected trait is rearing ability. So we do actually have separate contemporary groups for those. For yearling ewes, we actually account for the month of the year that they were born in, and the stage of the dam, whether their own dam was a yearling or an adult ewe, because we know that that affects the performance of those yearling ewes. And for 2-year-old ewes, we definitely accommodate the previous status of that ewe, because we know that it affects the reproductive performance of those. But for adult ewes, we're not going to correct for the status on a year-by-year basis, because you're chasing your tail if you do that. We have repeated records for this ewes, so if she has not conceived within a year, then that will be giving her negative results for conception, and the litter size will be solely based on her litter size records, for example. Then we actually do those contemporary groups within flocks, so that if people are managing different groups, you know, differently in terms of age, then that becomes part of the flock solutions. So it allows the effects to be different across flocks. We then estimate the other effects directly from the data, and these tend to be trends or patterns that are common across flocks. They might be a little bit different between flocks, but mostly they're fairly common in magnitude. And that's things like age, the birth rear type grouping. So here I've just got singles born and raised as singles, singles raised from a multiple birth, and multiples reared from multiple births. And we also account for the litter size challenge for the rearing ability trait. And we can do this, we haven't been able to do this in the past, we've had to do pre-corrections, but one of the developments I mentioned earlier was the software developments, and it's called RaceMule. And it enables us to actually estimate these effects directly from the data at the same time as we're producing the breeding values, and at the same time as we account for the other contemporary group information that's in there. So the first thing, of course, we did was test to make sure some of these corrections that are being applied are actually sensible looking. And so I've got a series of them here, and I'll probably just whip through them because they're not key to your RBVs, but it's just to give you confidence that the system actually, you know, does detect these effects well. So this is just an example of the birth type, rear type effect on various weights. And I put this here because I think everybody's familiar with the effect on young animals, but perhaps they're not so aware that the effect carries right up to the pre-joining weights of an adult ewe. So you can see that even as an adult ewe, a ewe that was born as a single and reared as a single has a weight advantage over ewes that were born as multiples, reared as multiples. So that's a multiple reared as a multiple is basically sitting on 0, which is why you can't see it. And similarly, I just want to mention that these early development traits that we're using to work out whether there's going to be any impact on the ewe reproductive performance, we correct these for age. So this is a bit different to the main analysis where perhaps these, you know, are not. And what I'd like to have you think about is that each of these kind of reflects a growth rate. So if we get an age difference equal to, this is about 160 grams. So these are all in kilograms. So that's 160 grams. You can see from this graph why we're quite keen for people to actually have correct dates of birth, because 160 days, 10 days difference here, 120 grams a day, and you've got more than a kilo's difference that's just due to age and not due to genetics. And this is part of the reason why we would be correcting data for age. We actually want to see the development of an animal on an age constant basis in this particular analysis. Again, this is the birth type, rear type effect on development. And you can see that it doesn't have a lot of impact on carcass fat, but it's certainly the muscle pattern actually follows the weight pattern, and it's also evident in the scroll measures. And again, the age regression. So, you know, knowing the birth type, rear type accurately for an animal and knowing its age are pretty important things. So how does that move on to reproductive performance? Well, it's actually not something that we fit for adult performance because there's too big a time difference between those two things, and we might actually remove genetic merit by doing that. But you can see that a singles born lamb actually has a bit of an advantage in terms of its performance for litter size and conception compared to a multiple reared lamb, even though the genetic merit might actually be the same or worse. And we can also see a small effect on condition score in adult ewes. So I've done these graphs separately because they're in different units. So we also correct for age of dam in our models for all of the indirect early development traits. I don't know if these have been shown elsewhere from the main analysis, but certainly you can see here that a 2-year-old ewe, which is basically on the 0 line, everything's shown relative to that, and here's the age group colours. You can basically see that your 4, 5, 6-year-old ewes have got your peak performance in the development of their offspring. So we do all of these corrections to use this data for the correlated traits. Now, as far as the ewe itself goes, her own age, of course, is also important. You can see the ewe age effect here on her pre-joining weight. So peak weight in Merinos is at 5, 6, 7, that's actually a year after the maternals. They tend to be a year earlier to hit their peak. The condition score on this graph, I think, really just reflects the fact that we don't have enough condition score data yet to show the ewe age effects. But if I show you what we have in maternal ewes, you can see that the 4, 5, 6 group have the best condition scores, and they start dropping off in condition score after that. And I would expect to see the same in Merino ewes when we've got more data. As far as the ewe age effect on reproductive traits, you can see again that the 2-year-old ewes, which would have everything sitting on 0, are the worst performers. And then by the time we get up to 5, 6, 7, that's when we're seeing the peak litter size for Merino ewes. And probably also of interest is we're getting a litter size, a slight reduction in litter size up here, but probably what's worse is that we're actually getting losses in conception, and we're also getting more lamb losses as the ewes age. So it's important to do all of these corrections so that we don't confuse, say, the observation of weight with the observation of performance, which is actually age-related and not genetic. Finally, the maternal behaviour score. You can see that older ewes have better maternal behaviour scores, but once they get to about 6, they're not really changing much after that. So that's really just to give you a little bit of confidence that the data that goes into the system is showing sensible results for the effects that we have to correct for. We've then estimated all the new genetic parameters from the data and the models that we've applied. And I haven't put all of them here because you don't really need to see all of the heritabilities for things like weights and fats and muscle depths. But this is just to illustrate that these developmental traits that we can measure early on, they tend to be, moderately to highly heritable. And so does the pre-joining weight. Condition score is about 0.2 heritability. Maternal behaviour score about a bit below 0.15. We have a really nice heritability for whether ewes will conceive as yearlings, and that is a lot to do with whether they achieve puberty by the time that they're actually joined. And then we have the lower heritabilities that we would expect for the reproductive traits of adult ewes. A bit lower probably than maternal breed ewes, and that probably reflects the use of more scanning data in this data set and probably more diverse environments as well. Now, all the correlations between traits, it's probably not a good idea to try and show you lots and lots of numbers for every combination of traits. We have to fill in a matrix, several matrices with these correlations that come from the data. If I can find my arrow. Here we've got, down here we've basically got all of the development related traits, and you can see that the stronger colour means that these are more strongly related to each other, and that's exactly what we expect. We expect the development traits like weight and fat and eye muscle depth to be related to each other and also to be related to condition score. Here we see a strong block of colour which shows how highly correlated yearling and post-weaning scrotal measures are. And then up here we have another block that shows that litter size and yearling litter size, where is that arrow? Here, are quite strongly correlated, but yearling conception and adult conception are less correlated with that. So probably the important thing to notice is if this is the block of reproduction and this is the block of early development, then this is the block that represents all the correlations between reproduction and early development. And because they're paler, that is showing you that they're all basically weaker than within each of these trait blocks. So there is information there. There are some favourable correlations there, but they're not as strong as, say, between reproductive traits or between condition score and the other development traits. So the key information I want you to get about the correlations is that actually at the moment the yearling information is pretty limited for Merinos, but we do have parameters and we are confident that the breeding values we're giving you are fairly good. Weight, condition score, fat and muscle depth are strongly correlated across all ages. So if you have those data in the system, you will get good breeding values for condition score. So plenty of accurate data for RBVs for condition score. Then we have the body development traits, which have favourable correlations with basically the reproduction and new behaviour. But most of these are actually fairly small in magnitude. Remember, we've removed all the non-genetic things that you might observe that might create perception of certain relationships between traits. With those magnitude of correlations, it still demonstrates that you need to actively record reproductive data to attain accuracy in breeding values for reproduction. And we can also use the scrotal measures because they're well correlated with conception and litter size in the female siblings. And just the final feature of the analysis is, as I mentioned earlier, that this is a single-step analysis. It's the most advanced way of using genomic information in analyses to produce breeding values. And Andrew's actually going to talk about that shortly. So we'll stop again. We've covered the models a bit, and have we got any questions?
I think Andrew's doing a pretty good job at covering off on those in the chat. Please let us know if we haven't covered off well enough there with what you're asking. But Kim, I did have one question now that we've gone through the data and looked at the different heritabilities. What would you say are the key differences, having done this for maternals and now for Merinos, between the data sets and the findings you got out of it?
Well, we're actually going to redo the maternal ones because we've got more data in maternal ones. But I would say that as a general rule, that early development is probably a little bit more favourably associated with reproduction in Merinos. That there is a kind of more consistent, perhaps, relationship between the ability to grow and develop well as a young animal and the reproductive performance as a ewe. But, you know, I might have to bite my tongue and revise that once we've re-estimated the maternal parameters again. So, you know, these things are, when we first estimate parameters, we need a lot of data, and sometimes it takes a bit to come down to the final set that we want to use. But we're pretty confident these are good ones in the current data that we have.
So there's been a few questions come through. One was that Andrew has partially answered, but around around the management. Sorry, I'm just finding the question myself. The post-weaning management of ewes. So after you've weaned the lambs, can you explain the way to manage back to joining? Is it good to have all ewes in the same mob, or should we be splitting on condition score? And what difference, you know, would that make? And accounting for them all in the one management group.
Well, I guess I'm probably not a management expert. So this is part of the reason why things should be fitted on a within flock basis, which is what we're doing when we fit contemporary groups within a flock instead of doing pre-corrections. So one flock, for example, might choose to manage ewes differentially in order to try and get all of the ewes back to the same time point, you know, the same condition pre-joining. And another flock may choose to just let them sort it out amongst themselves. Those are two very different philosophies, and they probably won't give exactly the same results, but it's not really our place to try and make people do things one way or another because that's their business decision. What will matter is that the contemporary group solution for that flock might be different depending on whether they choose to do it one way or another. And so from a year-to-year change, you might change your management from one year to another. That will be picked up as the contemporary group solution for those animals, and it won't impact on their assessment of genetic differences, for example.
So define your groups well.
Yeah, define your groups well. If you treat animals differently, you really should tell us that. If you treat them the same, then you know, put them in the same contemporary group.
Very good. Thanks, Kim. And thanks, Todd, for the suggestion there too. Admittedly, there's a little bit of chat going on in the chat box, which is really good. So just to briefly summarise, I think that pre-joining question or management, sorry, post-weaning to joining was one of the main conversations. And the other one was around the impact ewe weight change with ewes that are joined as lambs and how that impacts the data. And Andrew summarised that nicely and said we actually don't have a great deal of data in Merinos to look at this at the moment, but we've taken that question on notice to look at that a bit better in the maternal data.
Yeah. So we do know from the current data that based on one of the parameters that we actually have to estimate, we do know that ewes that are consistently high performers from a reproductive point of view are going to definitely suffer on their condition score. So they'll tend to come in a bit lighter at the moment, you know, like with a lower condition score. So it's called a negative covariance between the two traits within ewe. And we do see that. So we certainly know that exists. The management issue, again, though, I say is it's a bit of a business decision. And I guess I'd be managing the ewes in the way that, you know, optimises the chance of all ewes conceiving and lambing with a good result the following year.
Very good. And there was a couple more questions just come in from Mark. Is the 70-day minimum for preg scanning from the start of joining or the average?
No, so that's applied at an individual ewe level. The reason we've chosen 70 days is because if you have a 35-day joining period, 70 days after the ram end date is actually the minimum age of the fetus is now 35 days. So if part of the difficulty we have is that we've insisted on a ram in date, but we haven't insisted on a ram out date. I will talk a little bit later about how you know, people might kind of work that out from their ram out dates as well. It's just really to avoid the situation where the that individual ewe has been scanned so early in her pregnancy or so late in her pregnancy that you can't actually reliably establish conception or litter size from that scanning event. So we do actually use it still for conception unless it's too early.
Yeah, good point. And there was just one more come in, which Andrew has answered around the Merino parameters that have been established. Are they based on robust numbers and are they a good representation? And I think, Andrew, you'll touch a little bit about this on the when you're talking about the reference data set, but and where that information's come from. But so yeah, they're based on the entire data set. But Kim, did you have anything else to add there?
Well, I would. I mean, you're pretty lucky to have 250,000 and 500,000 records to estimate parameters. So yes, they are very robust for the population that's actually being put through the research breeding values process, and they reflect a large number of flocks and a very large number of size and contemporary groups. So I'm certainly happy that they're relevant to the population that we're actually putting through for the RBVs.
Excellent.
Yeah, and from a statistical point of view, the standard errors on those estimates are very small. So that's indicating reliability of those estimates on average for that population.
Very good. And I think that's a good segue into this next section, Kim, on testing these RBVs.
Yeah. So, I mean, we obviously do test breeding values because anybody can produce numbers out of any data set or anything. And the question is, are they the right numbers or the wrong numbers? So we do test our systems in a variety of ways. So I'll just outline briefly how we've tested these this time. We took the data from 2000 to 2014 in order to estimate the RBVs for the traits in question. We've used all of the pedigree and all of the genetic groups that are typical of the Merino analysis, and we've taken the new data and models in Solver, and I've just talked you through some of the systematic effects to show that we are getting sensible estimates of systematic effects from these data and from these models. So that should give you confidence that the models and the data are pretty good. But then we actually test the predictions of the breeding values, and to do that, we take the daughters from 2015 and 2020, and we look at their performance relative to the research breeding values we predicted on their size from all of the earlier data. And that way we can independently say, well, we had some breeding values on some size, so did the performance of their daughters, which were not used to calculate those breeding values, did the performance of those daughters then match up with what we expected to see based on the sire breeding values? So the correct number to get if everything's perfect is 0.5, and I've only shown you the ones here where we actually have enough data after 2014 and enough data before 2014 to get accurate breeding values for size in the first place and on enough size, and then have enough daughters after that 2014 break to actually do the daughter performance thing. And you can see that for 3 of the traits where we do actually have enough data to do both these things, you can see that condition score's almost spot on. Conception's almost spot on, so this is the standard error of the estimate. Litter size is absolutely spot on, and lamb survival, we're just doing a bit of an over prediction at the moment, but that also is a more problematical trait in the first place. So it's because it's less reliably reported. So these are really, really excellent results for the predictability of sire breeding values for their daughter performance. If we look at percentiles for the 2018 drop, you can see that I've just taken the middle out of these percentiles and reminded you what units the phenotypes were actually in, because we're not doing any multiplication or anything when we give you the results. We're presenting exactly in the range that we've actually analysed the data in. So this shows you that the difference in RBV for a condition score is from -0.43 to +0.7 in condition score. So that's a range of close to 1.3. This is yearling conception and adult conception. You can see that it ranges from, you can think of this as almost as a percentage, so +14% to -13% around the mean. The litter size, this is in lambs, so 0.37 lambs to -0.27 lambs. And you might wonder, how do you interpret a number like this when lambs only come as wholes, they don't come as part lambs? And really this is probably the easiest way to think of this is if you have a 0.1 breeding value for litter size and you have 10 daughters to compare for 2 rams, then one of them will be having an extra lamb for the litter size that is 0.1 plus 0.1. That's probably an easier way to kind of think of that concept. And then you can also think of era as a bit of a percentage as well, because it's a division based on the lambs born. So you can see nice range here in the RBVs for lamb survival and also in the maternal behaviour score. As far as reportability goes, because we're not you know, breeding values is not about throwing all data at a system. It's about throwing correct data and useful data into a system. So ideally, we would have a fairly high percentage of reportability when we do that, and that is indeed the case. So if we look at the size of progeny in 2018, between 65% have reportable breeding values for maternal behaviour scores, up to 78%, 77, 79% for condition score. So a lot of sires are getting reportable breeding values, and the percentages of progeny that are getting reportable breeding values are also quite good. The genetic trends, don't forget these genetic trends are based on historical selection for number of lambs weaned, which is actually a less accurate phenotype than each of these phenotypes. And you can see that based on previous selection for NLW, there's been a very small positive trend in conception and lamb survival, but by far most of the trend in NLW has been driven so far by litter size. And that makes sense because litter size is the more heritable of those traits. With respect to the improvement in accuracy resulting from the genomic information, the maternal sheep breeders were fairly disappointed to see no significant improvement, but in the case of Merinos, we're getting somewhere between 5 and 10, a little bit more sometimes, improvement in accuracy due to genomic information, and Andrew will talk about that shortly. So if I just summarise the overall thing for the new RBVs, we're basically focusing on maternal performance traits, and they're in components, so they're not a conglomerate trait which can have an unclear meaning. We use improved data, and we've increased volume for some of that, quality, consistency, and we've improved the usefulness of multiple data sources. We also now have better models. We make better use of the correlations between component traits and any other trait that we want to use as an indirect indicator. And we use the single step method, which is basically the Rolls-Royce of using genomic information. And therefore you will get new research breeding values for conception, litter size, rearing ability, maternal behaviour score, and condition score. And we're able to demonstrate that those research breeding values currently have good predictive capacity, and I only expect them to get better as people improve their recording and we get good recording across a broader part of the industry. And although Andrew and I and Emma are doing lots of talking, there's actually quite a few people at AGBU who've done a lot of work on getting these RBVs in place. So I'd just like to acknowledge all of those. And we'll stop there, and that will give a chance for a couple more questions before Andrew talks about the genomic evaluation.
We haven't had any more come through on the chat, but feel free to type anything you might have. We will come back to Kim after we hear from Andrew. Kim, I just had another quick one. You've spoken about these traits having a slightly lower heritability, but if you were a Merino breeder and you've got these new RBVs, how would you go about making gain in these traits?
So people often confuse low heritability with an inability to make change. But when you have a lot of variability, and that's part of the reason that we show the percentiles, it is definitely possible to make change despite a low heritability. So I think Emma's going to talk a little bit more shortly about how people can actually start thinking about using those research breeding values, but certainly don't let the heritabilities put you off, because if you record it and you've got that information there, it's certainly possible to make change, and there's plenty of industries that can demonstrate that.
Great summary. Kim, there's been nothing else come through. I'll hand over to Andrew now.
Okay, we're going to do a quick swap.
Thanks, Peter. Thanks, Kim. And hi, everyone. So I was just going to give you a little bit more detail about the genomic side of this new analysis. As Kim said, it's a single-step analysis. That's just a single step. It's just the way we refer to the way we calculate breeding values these days in an analysis that includes all information at once. So pedigree, genotypes, and the actual phenotypic data, which in this case are reproduction and associated traits. So that's what the single step means. We've been doing it since 2017 or 2018 for the main analyses, but we've never done a single step analysis for reproduction in Merinos, and that's mainly because we haven't had a good enough reference population to underpin the genomic predictions until now. So I'll get onto that in a minute, but just to highlight the point which Kim just made, and that's the graph that she showed. What that graph actually is the difference in accuracy for flocks who have done a lot of genotyping on their young rams. And what we actually see is if we compare the young rams in those flocks that have been genotyped to their contemporaries that haven't been genotyped, those young rams get an increase in accuracy, a boost in accuracy due to having the genotype of between 6 and 12 accuracy points across the range of traits. So all in all, if we have that increased accuracy in young rams, that translates directly to increased genetic gain when we're selecting on these traits. So that's why single step is such a good thing, and the use of genomics, and it's been really pleasing that we've seen that increase in this analysis. Just to explain a little bit about how the genomic prediction actually works, if we just genotype animals, that might not necessarily do anything unless we also have phenotypes to associate with those genotypes. And that's what a genomic reference population is. So for the genomic reference population, we need to record reproduction on traits of a lot of ewes, and then we have to genotype them so that we can make a link between their the pattern in their DNA, in other words, their genotypes, and their actual performance data. So
Andrew, sorry, before you go on, you've got to, just had a couple of people say your audio is a little bit shaky. Would you mind maybe just sitting in closer to the microphone might help? Sorry.
Okay, so just interrupt me if it doesn't work so well again. So the next step is that a breeder in their own flock would DNA test young animals, and then would be able to use that link to that genomic reference population to make a more accurate breeding value just by virtue of the fact that they have genotyped their animals. Of course, in single step, it includes all the other information on pedigree and performance data as well. So we've got everything happening to increase the accuracy of breeding values. One important thing is that your relationship to the reference population matters, and really the way that is actually addressed is to make sure the reference population is representative of the whole analysis that you're trying to provide genomic information for.
I still think we're having a few problems. Is your other computer on mute, Swanee?
No change.
No change.
Okay, we've shut that computer down. Just trying to turn my volume up. So,
it's not a volume problem. I think it's might be a microphone. The quality of the microphone might be an issue.
It's the same computer as I just talked through.
Yeah, you sounded fine then, Kim. Doesn't like Andrew.
It's my computer, so it's probably saying,
Okay, well, we'll just have to press on. I don't really have that much more information really to share with you. But just to say that as far as reference populations go, it's taken a while to build up the reproduction reference, because you do actually need a much larger population because these traits are such low heritability. The other thing is that breeders typically genotype their young rams. That's where you get the biggest bang for your buck. But what you actually need to do is to have the phenotypes come from the ewe side. So we need to genotype ewes. So the reference population will not naturally build up in breeders' flocks. We need to do some strategic investment to create a reproduction reference. So there have been several initiatives to do that. The first one was the information nucleus flock from the sheep CRC, which ran from 2007 through to 2011. So there was a limited amount of reproduction data created in that on Merinos. The next one that we've seen is that we've had some strategic genotyping actually in Merino breeder flocks, but that's where the MLA reference flock project has gone out and invested in genotyping females in those flocks. And the last one is AWI's Merino lifetime productivity data, which is now in the Merino Select analysis. So genotypes on all those ewes out of that project are now in the analysis. This is just to show you how the numbers have changed over time. You can see that INF animals in the early days, there are just over 2,000 ewes genotyped with reproduction data there. And then the Merino Select, in Merino Select flocks funded by the reference flock project coming through there after that, and also the MLP data in recent years. So that's contributed, those have both contributed over 3,000 ewes. Now, what we need to do is ensure that you know, there are more ewes coming in behind those in future years to underpin the reference flock. This is just to show you that it's another way of looking at the boost in single step. You can see, for example, litter size there, the accuracy of predicting future performance. That was part of the same validation that Kim spoke about before, but you can see the boost there that we're getting for litter size of a single step compared to just a pedigree model. And the graph on the right there just shows a theoretical relationship between the accuracy of genomic predictions on the y-axis and how big your reference flock actually is. We've got lines there for traits with different heritability, and you can see at a lower heritability, you can only expect a lower level of accuracy for the for a reference population of a given size. So, but pretty much that lower line at heritability of 0.1 is about what we're seeing for the increase in accuracy for litter size. The other traits have a lower heritability, so they're a bit lower. But you know, what we're seeing in this analysis is actually lining up very well in the case of litter size with what a theoretical prediction would say. In order to increase the accuracy that comes through genomics, what we need to do is increase the reference population size. So that's what I mean by we need to continue looking at how we do the best investments to improve genomic accuracy and have that flow through to improvements in accuracy on breeding values for individual animal and increased genetic gain. So just to summarise that, as I've said, the genomic information improves accuracy, and with that we will get an associated increase in genetic gain, but we also need to plan future investment in reference populations for reproduction. So that's what I have, and I apologise if my sound wasn't very good. Maybe I should let him do that.
Thanks, Andrew. Has anyone got any other questions for Andrew? Just type them in the chat box. There was a little bit of discussion there around, you know, talking about genomic reference populations. Is it just DNA parentage or is it a full genomic test? So we clarified that it's not just DNA parentage. It doesn't contribute at all to our genomic reference populations. It needs to be animals that are measured for that trait and have a full genomic test done.
That's correct.
Feel free, if you think of anything to ask Andrew, type it in the chat box, and if not, Kim, I'll hand back to you.
Okay, thanks, Peter. You're probably all sick of hearing from me by now, so I think it was good that Andrew actually talked. Emma and I are going to cover a little bit about recording for reproduction. I did see the tail end of Ferg's comment on scanning, so I'll cover that a little bit more now. I'm just going to focus on 4 things because in developing our software for, you know, sorting out the data, we've seen some pretty common problems. So I'm just going to focus on a few problems, which are probably the main contributor to people not getting data in. And I'll just summarise these quite quickly. We are actually building a lot of data off, you know, information provided by breeders that don't use the mating module, providing they meet our other filters. But essentially, the real problems occur when basically people provide not dams for lambs, or they don't provide all lambs for the dams. So we basically need dams for all lambs, whether those lambs are living or dead. And ideally, people would supply data through the mating module. There's a definite issue in most data about having the correct dates for different things and the correct timing of scanning on all ewes within a contemporary group is also an issue. And I'll just cover each of those bit by bit. So essentially, to have you know, your rearing ability traits, you must report dead at births. We don't currently have a way around that. And this is because we need the consistency between birth type and the lambs identified in the data, and we need it for the rearing ability. This can actually be a problem when people rely on DNA parentage because they often don't supply dead at births. They might have full pedigree, but they don't actually supply dead at birth, so they end up not having any reproductive data for lamb survival. And this has become a bit of an issue, I guess, more for merino breeders, but we actually need a bit of a push from breeders to the software providers, so for your herd recording, to actually work out a way of supplying the dry and dead at birth tags for analyses, because we can only use what we receive, and we definitely don't want to be creating phenotypes, you know, based on the difference between birth rear type and scans, when we know there's errors in rear type, we know there's errors in scans, and it really has to be the breeder who validates the fact that there has been a dead lamb or a lamb that wasn't reared. We don't want to generate that for a bunch of flocks. It may be correct for some flocks, but it may be terribly incorrect for others. So I think this is a bit of an issue of taking note of the software that you have and the capabilities of it, and working with the software developers to actually get that right. And we know also that the tiered flocks is a bit of an issue. For example, a tiered flock might appear to only have 50% dams recorded, when in fact they're recording their entire nucleus of that flock. You know, the information's being eliminated because only 50% of dams were reported. Now, ideally, we would be able to have a way to work around that, and I know that sheep genetics will be working on a way that we can somehow identify that these are the ewes that we should actually look at, and not all ewes, for example. The second thing is really to supply data through the mating module. So People are interested in condition score, maternal behaviour score, etc., and getting their pregnancy scan data used. And we're only accepting that data through the mating module because it's then tied to the joining event and to the actual lambing event, and we know that the data is correctly aligned with the outcomes for that joining in that year. And it's also the place where you can tell us about the joining groups for ewes. There's no way of us actually establishing joining groups for ewes from the data that's based on a lamb level accurately. And this gives you the opportunity to actually tell us that these ewes have been in an ET or an AI program, even if they then conceive to a natural backup lamb later, they still should be listed as being in the program where they had pharmaceutical kind of interventions. So this is really the only place where you can tell us about joining groups for use. And, you know, you can create those joining groups in a meaningful way for you. That's, you know, that might be the timing of the ewes going into the rams differs. So that might be multiple joining events in a year. It could be that this group of ewes have been given 80 days to conceive and that group have only been given 35. It could pick up health issues or sire failure. So that's the breeder's opportunity to tell us which groups should be compared and which groups should not be. And most breeders will actually only have a few groups, so it's not too difficult to provide the joining groups. And if you're confused about how to actually define a group, then you can always talk to us or talk to Sheep Genetics, and we can work through different scenarios. We're using dates a lot to check and validate data, and there are some unusual dates provided for outcomes. So what we really would like is in that mating module data for people to tell us what their in and out dates are correctly, definitely be correct on the scan dates. And a lot of the lambing dates too don't tend to be particularly accurate, so they might be spread out, you know, reflecting weeks, or they might even reflect an average or just a couple of time points throughout a lambing interval. And because we use those dates for actually checking and correcting, it's better if they're as accurate as they can be. So things like dead at birth and dry lamb tags should be within the range of lambing dates for all of the lambs that are part of that outcome from joining. And supplying correct lambing dates as possible is ideal to help us improve the filters on the data and make sure our checks are all pretty robust. The correct timing of scanning on all ewes is an issue. You've got to remember that using an average to try and decide when the scanning date may not be very useful. And also altering pregnancy scan data to become birth type or vice versa is also not very useful, because it means we can't tell you about the accuracy of the outcome. So if you talk to scanners, they generally have a setup where you can tell them joining dates, but they probably don't actually consider the length of the joining period. An easy rule of thumb is that you basically shouldn't be scanning ewes any earlier than 35 to 42 days after the last date that they were exposed to rams. And if you want to keep them in that window of 35 to 110 days, then that would mean that the length of joining can't be more than 60 days for a single scanning event. And the more you push this up, the shorter this has to become to keep all of the ewes within the right scanning fetal age range for scanning. And we want to receive the first scanning result, with, you know, the one that's relevant to the first opportunity for those ewes to join. So if you're concerned about ewes being compared that were in an AI program because they didn't conceive from the first joining, which was actually their AI, this is why we're telling you to tell us, is the ewe in an AI program or not? Because they'll only be compared to other ewes in the AI program if you can tell us that in your grouping. And finally, I guess we always provide a lot of diagnostics to sheep genetics to help breeders, and sometimes maybe it's a bit off-putting to look at some of these diagnostics and go, well, there should have been 400 ewes there and there's only 10. That's going to take hours to fix. Well, that may not be true, because some of the exclusions are based on groups. So it might be a matter of fixing 10 or 20 ewes. I mean, you forgot to put in dry tags, or you forgot to put in something else, and all of a sudden the whole 400 slip in. So it's really a matter of working through those diagnostics with sheep genetics and work out, is this an individual ewe problem or is this a group problem? And the group problems can actually be quite easy to fix. So there are a number of exclusion reports provided, and Emma will actually talk more about those. And that's about all I'll say for the time being. I think it's probably better if she works through a bit of an example with you. Lots of questions here. Lots of questions. Okay.
Yes, lots of good questions come through. And before we go to Em to talk about interpreting reports, one that came through was, do you feel that the challenge for rearing lambs from a litter where there were multiples has been accurately accounted for?
So the solution is 10% reduction. I would think that the solution should be bigger than that, and that probably reflects the fact that we are a little bit lenient with the data coming in, as far as only insisting on an 80% consistency of birth type with lambs reported. If we insisted on 100%, we'd get a lot less data, and we might actually get a nicer, clearer result for the impact of litter size on lamb survival. But certainly in the data that's generally available, for singles and twins, the most common result is for the ewe to actually rear the litter size that she had. So even twins are more likely to rear twins than they are to rear singles, for example. By the time you get to triplets, that switches over, and that clears up. It shows up in our solution in the correction for the challenge. Now, I think that solution will get bigger and stronger as we get more data and more accurate data. But at the moment, the triplets are probably about no more than 5% in most populations. In fact, in most populations, the percentage of triplet litters is very low. So for people who have a lot of litters, maybe a stronger correction would be better. We've proven that in other work, but for the national evaluation, it's pretty hard to actually make it exactly the same, level of integrity. Yeah. So that's actually an issue for the industry in a way. The challenge, we always have a trade-off between how much data we can get in that will give a broader coverage to the industry with breeding values, and they'll be pretty accurate, versus how stringent do we become to have the highest heritabilities and the maximum response, but in the fewest breeders' flocks, because only those flocks that record really accurately would get in. So at the moment, I think we're kind of somewhere in the middle. For people who have really, really great data, and I think there was a question asked about this earlier, if people had really great data and they had a very prolific population, maybe there would be some interest in them having a similar analysis, but at a higher level of stringency and probably higher heritabilities.
Yeah, thanks, Kim. And I think, Rick, that answered your question from before around if we've looked at the different heritabilities for different flocks. We had one come through from Caitlin, which is a really good question, because we often get asked about it a lot when we're out and about. It was around if there's a really bad weather event, for example, how that you didn't want to attribute it to the ewes, how would you go about treating that, Kim, when you're entering data?
Oh, you know, that's a great question. I can tell you the way I'd like to treat it. If people could give us accurate dates of birth, I could tell you now that we would easily spot weather spikes that, you know, into lamb deaths. The big difficulty is that people are not giving us those dates, and so we're unable to use an automated method like that to actually identify those lambs. The second answer is we're already splitting up lamb groups. So in the ERA, we're I think it's about 20 days per group once the lambing period is over about 60 days is the minimum size, and then there's up to 4 splits within a lambing group that we're automatically putting into the model. So hopefully the weather event would be confined in one of those splits, and that will help a bit with that particular issue. It's the best we can do without actually having the lambing dates. The other issue, of course, we had was that we were using people's group descriptions about lambs. But what we discovered was that a lot of the group descriptions actually split up contemporaries. So, you know, a male sibling of a litter might be called, you know, male nucleus lambs, and a female of a sibling might be called a female nucleus lamb. So the description that we could have potentially used, which might have said weather event lambs or something else, has been used for people's convenience to kind of look at groups of animals that isn't related to, you know, splitting those animals up for a genetic analysis. So we haven't been able to have a nice, clean way of people supplying information on actual contemporary groups for lambs. And that's why we've resorted back to the splitting option. And we have been in discussion with sheep genetics about how we can try and get better information on lamb contemporary groups. And that would be where you would put your weather affected dead at births.
Yeah. So in summary, Kim, and we've had a couple more comments or questions come around what you were just talking about. So for people at the moment that are doing date of birth or wanted to enter a status, we're not using that in the RBVs, but we would encourage people to do that in the future so we potentially can. Is that an accurate description?
Yeah. So really these things come down to, again, it's this question of broader use versus more stringency. So, you know, one person can do a brilliant job recording everything, but because we can't use it in a robust fashion across everybody's flocks, it tends not to get used. If the industry were to move towards more consistent recording in how we can actually identify these groups that should be excluded, and we have done some work on that with sheep genetics. There are various codes. I know they're a bit complicated and stuff, but there are codes that could be used. When we get enough information from that, we then definitely do look into whether we can actually improve how we split up groups of animals for that sort of thing.
Very good. And just quickly, I'm conscious of time and we've still got to hear from Em, but there was one comment. Would you mind just clarifying with some of the data, making the most use of your data? Someone commented, does that mean we have to DNA dead lambs if they're using parentage? Did you want to just clarify that, Kim?
Oh, yeah. So of course people aren't doing that and they don't want to do that. This is why I made a comment about breeders discussing with service providers, software service providers, with respect to herd recording, how they can identify those dead at birth lambs. Now, if the breeder was happy that their preg scanning was accurate and that they wish to use the difference between the preg scan and the, you know, the lambs reared as the source of how many lambs were born dead, to that ewe, then that would be something that they could provide to us as a dead at birth tag if they set up and validated that system with their software developers. Now, we've been asked whether we can do that ourselves, and I mentioned earlier we don't wish to do that because for some people it will be very right and for some people it will be very wrong, and it really should be up to the breeder to validate that this is the way I believe my data is correct, that there was in fact a dead lamb born to this ewe. So that's how we would prefer it to be. I've also talked to the DNA company saying, you know, it would be really great if you could DNA dead lambs for a reduced price. And maybe that's another thing to take to the companies that provide DNA testing, whether, you know, they can start offering a you know, 1 dead for 10 live deal or something like that. It's a hard one to overcome.
Thanks for clarifying that, Kim. There was just also a bit of discussion around entering data via the mating module. So thank you. I think Sally clarified that question. Now, Kim and Swanee will stay on the line, but for the next and final session, I'll hand back to Emma.
Okay, so I'll stop sharing now.
Thanks, Kim.
Does that look okay for everyone? Yeah. So I just wanted to touch briefly on what you guys will actually see. So the way that we will report these research breeding values back to you. So we currently have these new RBVs available on the search website. They will be highlighted in purple. That's just to reiterate that they are research breeding values. And there will also be an additional report on your analysis results email, which will be a PDF report, which I'll go through in a bit more detail. These will be sent out this afternoon. So this catch up will still be fresh in your mind, hopefully. So this PDF report has 2 components to it. It has a data count as well as an individual listings. This first page, the data count page, shows you how many ewes have been recorded for each of these traits. So for conception, it should reflect a count of ewes joined. For your litter size traits, it'll be those pregnant ewes. And for ewe rearing ability, it'll be ewes that have lambed. We've then also got a count of your condition score records and your maternal behaviour score. It's important too that each of these are based on the year of joining rather than the actual year the dam herself was born. So it'll be a count of the dams for each of those years. Yep, there we go. So this report's really useful for identifying gaps in the data. So as Kim was just saying, individual ewes and groups may be being excluded. So it's a good tool to use, and we can help you with using it to identify any missing parts of your data. So the first trait, conception, the count there should generally reflect the number of ewes that you've joined. There's a few common exclusions we see, so things where you've got really high conception in a group, so we don't have any dry information, or we have a service sire failure where we don't want to be penalising the ewe for a size mishap. The litter size count will reflect how many ewes are pregnant, or should generally reflect that. So it won't include your dry ewes there. The ewe rearing ability columns will then reflect the number of ewes that you have lambing. So it should be a similar number to the litter size count. With the ewe rearing ability, it's important too to consider your own flock mortality in that count as well. So if we actually apply it to a real example, This is a flock that will be receiving reports today. But it just highlights a few areas that, or a few of those gaps that we've been talking about. So the first thing when you're looking at this report is not to panic if there's zeros there. We need to consider a few things. So we need to consider that level of dam pedigree that Kim was talking about. So it needs to be at least 70% for that drop. We also need to make sure we've got things like our dates right. After that, we can then go through. So in this scenario, what jumps out here is we've got 0 records for conception for a few years. In this case, it was because we were missing dry ewe information on the entire drop. So we had no conception records come through. Another example here is where we've actually got a lower number of conception records than litter size and ewe rearing ability. The reason for this, it sort of highlights how we're doing exclusions at a group level. So we had a group of ewes come through that didn't have any dry animals with them, so they were excluded. However, there was other groups that were available there that had you know, a sort of realistic level of conception, so they were included. What we would normally expect to see is this trend here where we've got a higher number of conception records because we're including dry ewes in this count. And then these would decrease for litter size and ewe rearing ability because we're looking at the pregnant ewes and ewes that lambed. We're always available to go through this with you. So it's important for you to have a look at these and see if you can get to the bottom of gaps. But we're also willing to give you a hand if you're having any problems. And as Kim said, we can build these sort of, they're a manual exclusions report, but for those that are a bit difficult to get to the bottom of why we're not getting data coming through, we can do that to have a look at why they're being excluded. On this same PDF, you will then scroll down and you'll get an individual listings report. So that'll be similar to the individual listing you get already with all your normal ASBV traits, except that it will just be a reproduction RBV report. So we will get the breeding value. It will also be in purple, same as what we would see on the website, just for consistency. There will be an icon here to indicate if the genotype has been used for that animal. We have the accuracy below in grey. And then we have this tick or cross, and essentially the tick or cross is a linkage indicator. So it says whether your flock is linked for that trait and whether you can use it to compare across flocks or whether it should just be used for within flock selection. So what would a good RBV look like for your flock? This is where we use those percentiles that Kim pointed out earlier. This is just a screenshot of what you might see on the website. It's important to identify where you currently sit for these research breeding values, but then consider where you want to go with them. So the first step in that is deciding which direction you need to go. So for a trait like conception, you might be trying to increase it. And so increasing the breeding value would be the favourable direction. But we need to remember that with a trait like maternal behaviour score, a more negative value is actually more desirable. So we'd be wanting to reduce that breeding value. Once you've determined which direction you want to go, we normally recommend that you then use the percentile tables to identify what your target is and where you'd like to get to. So this is a maternal example. I haven't had time to pull out a Merino one yet, but I just wanted to show how you might put using the percentiles actually into practice when looking at your own RBVs. So this is a maternal sire. He's actually a pretty interesting one. He's been used pretty extensively throughout industry. And a lot of the reason for that is he has a really high NLW. He's in the top 1% of the maternal analysis. What the RBVs allowed us to see was actually what was contributing to his number of lambs weaned. So he had a really high conception. He sits in the top 20% of industry for conception. He also sat very high in industry for litter size, so he was in the top 1%. But he had an ERA that was in the bottom 10% of industry. So what the percentiles allowed us to do was actually identify what is a good RBV compared to the rest of industry. So that's all I wanted to work through. It'll obviously mean a lot more to you once you receive your reports. And I'm happy to work with you too to identify and diagnose why we've got gaps in data. And then we can work together to use the percentiles to actually provide some context around what those breeding values mean for you.
Excellent. Thank you, Ken. Em, if you wouldn't mind just stop sharing your screen so we can see everyone's faces. There's just a couple of really quick questions to go through. And I thought I'd just put maybe put a little caveat on comparing the RBVs to NLW. And I'm sure Kim and Swanee would probably elaborate more, but they are from very different analyses using different parameters. So to say, oh, this animal's really good for all these RBVs, but his NLW is low, it's not comparable in that way. Kim, I don't know if you wanted to add anything to that.
Well, all I can say is that from my perspective, we're being extremely careful with our data filtering, and for the majority of animals, there will be positive correlations. So you wouldn't expect a sire that's say very, very good for NLW to suddenly look terrible for all of the components of NLW. If that was the case, it would be, you know, a case of definitely pointing that out to us, and we'll go and actually have a better look at that animal's data. But by and large, the correlations are positive and quite moderately high. You wouldn't expect them to be 1, otherwise why would we bother changing, you know, from what we currently do to what we do now, which is a better job. So certainly the correlations are positive, but you might also have the situation where an animal might be unexpectedly worse for something when you thought he was really good, but that you have to break it all down and say, well, has he got as many daughters getting pregnant as another sire? Is their litter size better than another sire? And am I recording the lamb survival accurately or not? So definitely you shouldn't see anything that was good now turn absolutely terrible from this analysis, because it's more refined parameters, you know, to do each of the components better, but it's not, the underlying biology is no different. So, you know, we expect to see similar things.
Thanks, Kim. I know we're coming right up to the 2-hour mark and towards the end, but there was just a couple of quick questions that have come through that were really good around. The first one was, what's the next steps? How are we going to move from an RBV to an ASBV, and when do we expect that to be happening?
Oh, I might let Sheep Genetics answer that one. I mean, you know, we've got great predictability if the industry keeps on recording and keeps on improving their recording. There's no reason why we would not move these towards ASBVs. You know, we've got a pretty good handle on the correlations with the traits that we need. Probably the ones we would look into would be ones relating to wool. But certainly for the indicator traits that we've got, I'm pretty happy that we've got a pretty good handle on those. So usually the delay is with getting enough people having the data recorded and having enough confidence that the parameters won't change. But we generally update parameters as we get more data anyway. So, yeah.
I just want to give my perspective. I don't know whether you can hear me very well or not. I think this analysis is in many respects superior to the current reproduction analysis that we have. One reason to keep them as RBVs for a while is just to get people familiar with them and see what their acceptance is, so that, you know, we can modify various things about them. So, you know, it's a matter of just getting people comfortable with the new figures. Another job that we will have to do is to revise the indexes, the standard indexes that we have, to use component traits instead of NLW. So that really is a must-have before they go to ASBVs. I don't know if you want to add anything from Sheep Genetics' perspective, Peter.
No, I think you've covered off well there, Kim and Swanee. The only thing I would just add to that is that obviously before we move these on, we do have to get sign off by our technical committee and advisory committee, but mainly technical committee, to make sure that the work behind this is sound. Probably time for one final question. Or Rick, you just had your hand up. We'd love to hear from someone else other than the four of us.
Yeah, can I just ask the question, in the composites or in the maternals, they're a step in front of us by time. Are they saying, sorry, have they used these research breeding values into their indexes yet? How are they going with it? Where are they up to?
No, the index work hasn't been done for them either, Rick. So I guess they've had access to them for a year and they've been getting used to the figures essentially. But Merinos got done after maternal simply because the industry data is more difficult to handle. It takes a lot more work in actually developing the processes of getting the data together. And because you're using a lot more pregnancy scan in quite a few flocks in order to get your phenotypes compared to the maternal breeders who are doing a hell of a lot of mothering up. So, you know, that's the two reasons why there's that kind of gap, I guess, in the development, but the indexes haven't been done for either. We basically know how to do them because we know pretty well, you know, that as litter size goes up, lamb mortality does too, and you have to factor that in. So I think, you know, we've got a pretty good idea how to do it, and they'll probably get done in concert would be my guess.
There's an update to maternals too.
We're also updating maternals, Andrew just reminded me.
The RBVs.
The research breeding values. So we'll have another set of parameters for them and we'll update them again before we do the index work. Because we actually use the parameters in the index work as well.
Very good.
Thank you.
Glad that answered your question, Rick. Does anyone, before I do the last one in the chat, does anyone have any other questions that are on the conversation that wanted to unmute and ask? I don't see anyone coming off, so we're all available obviously after to answer any questions. But the final one, in terms of reporting, how do we report back on, I think in terms of the data summary this is relating to, but a ewe that was joined in December to lamb in autumn the following year. Kim, from an analysis point of view, that's that would be being handled?
I think I'll leave Andrew to answer that. We have a time check in our data that tries to distinguish whether people are doing multiple joining events in a year. If you're doing multiple joining events in a year, ideally you would be contemporary grouping those out as separate joining events. And therefore, you know, it really wouldn't matter what time of year you were joining them, as long as they're part of a management group that reflects that time of joining. If Andrew wants to add something.
Maybe just a small correction. This is a once we take one joining lambing event per year. It's really complicated to have more than that. But I think this question is about whether your joining happens in 1 year and it carries over to the actual lambing in the next year. Just have a look at your report when you get it, if that's your situation. I think that it may well stagger your the data that you've got into the next year in terms of, so the year of joining in that situation might actually turn out to be the year of lambing. But if it's confusing, just let us know and we will check it out.
Thanks, Andrew. That was all the questions that were in the chat. Final chance to ask anything. Rick again? Putting your hand up, that's very good.
Sorry, my wife just said, no, not you again. So when we download our sheep genetics updates, we'll be able to get them all. Have our so we're using our computer system. Are they updated with how everything's going to read?
Em, I'll pass that one to you.
I'm assuming you mean back in, like, downloading your breeding values back into your software, Rick. Yep. So they have all been involved in this conversation. So I mean, today will be the ultimate test, but they should all be set up now. No?
No, I from memory, sorry, Em, I think you'll get your reports if you haven't already. But in terms of softwares, because they're RBVs, with maternals, we've done the same thing. We haven't, they're not actually getting to softwares at this stage. It was just traditionally how we've handled RBVs, but we can do Excel files upon request if you'd like that. And if you think that it might be a common thing that you wanted in your softwares, it's always something we can take on board and discuss internally and talk with our software providers about.
Sorry.
No, that's all right. So if that's all for today, I know we've just gone a fraction over time, but we might call it quits for that. So thank you everyone so much for taking a couple of hours out of your day and joining us. We really appreciate you all coming along, being the people that are recording this data and will be using this data in your breeding decisions. It's been great to have you. Thank you to Em, especially for all the coordination around organising this webinar for you all. And obviously a massive thank you to Kim and Andrew. I keep wanting to call you Swanee. I don't know, Kim and Swanee, for joining us today, and the whole team at AGBU for the amount of effort and work that's gone into this. I really don't think to say a lot would be a massive understatement. So thank you everyone. And just as a reminder, the recording and slides will be able to send out for your listening pleasure, as well as a post webinar attendee survey. So if you could take 5 minutes to fill that out, that'd be greatly appreciated.
Yeah. So Peter, can I just say something? We've spent a huge amount of time trying to make sure we get things right, but I'm sure that you will, some of you will find something that looks odd. If it does, please tell us. That's the only way that we can go through and do any debugging if we need to. So, it can be at your end and it can be at our end. So please make sure you tell us if you see anything odd, either through sheep genetics or even back to us. And we'll do any investigation we need to make sure that we have indeed picked up all the permutations that are possible with Merino data.
Thanks, Kim. Alrighty, guys. Have